Well, good morning, everyone, and welcome to the APS Hot Topics, Everything You Should Know About CRISPR session. My name is Shaun Curtin, and I'm a researcher at the USDA-ARS, where I work on genome engineering of wheat, oat, and brachypodium. Over the last 7 years or so, I've been using these genome engineering reagents to work on soybean and metacargo and now wheat. And so I've seen the progression from ZFNs to talons to CRISPRs. And as I hope to convince you, these CRISPRs are quite a remarkable reagent. So the overview for today is I'm going to give a very brief introduction and overview of CRISPR-Cas9 reagents. And then I'm going to discuss 3 examples or 3 applications of how I'm using these CRISPRs for crop science or in crop science. And then I'm going to introduce two other speakers, Tomáš Čermák, who's going to introduce, or he's going to present his fantastic new multiplex genome engineering kit, which I think people in plant pathology will be very interested in. And then Diana's going to also present some of her work using this same engineering kit. So just quickly, an acknowledged slide of some of the people that I've worked with and collaborators who have been very helpful in this work, and also some funding agencies. Minnesota Soybean Board, USDA, United Soybean Board, NSF, University of Minnesota, and the Center for Genome Engineering, which is a center at the University of Minnesota. So these are the site-specific nuclease platforms. Their main purpose is to create double standard breaks. And over the last 7 years, they really came online. So we have the zinc finger nucleases. These in 2008, 2009, there was 2 platforms that were published or released, and they worked pretty well, but they were sort of a little tedious to work with, a lot of expertise required in engineering them. They were a little toxic to plants, and they worked 50% of the time, but at that time, that was pretty good. In 2011, the teleffector nucleases came online, and these were required some expertise to build. They also there was less toxicity. You could constitutively express them, and they had a much larger range of targets. So you could find a target one target every 10 base pairs. So they were really good. So you didn't have any problems like with the ZFNs. And then around about, I guess, 2014, these CRISPR-Cas9 reagents came on, and they've really changed everything. They're easy to make, they're cheap, they're inexpensive, and you can get them into the plant really easily, and they always work. That's the thing, that's the main thing. You don't have to worry about things not working. So just to talk a little bit about what this CRISPR reagent is. So we had some great talks on the Monday. It's an adaptive immune system from bacteria. I won't go into too much detail. It's got many components, but genome engineers really are just interested in 2 of these components of this adaptive immune system, and that is the Cas9 and this guide RNA. And you can see this is the target genomic region, and this is the part of the guide RNA that you can interchange. So you can change the sequence of that to target a different target. And this is a schematic of a plant expression vector, the tDNA of a plant expression vector, and this is how it's delivered to a plant. And so you would have a strong constitutive expressor, such as this one here is the glycine max ubiquitin. You have a reading frame of the Cas9 enzyme, which is this here, and that has a synthetic intron in it as well, so it just makes it a little more easier on the E. coli. And then you have a Pol3 U6 promoter, which drives this target and guide RNA. And then when you deliver those to the plants, they come together and they find their target. And as I mentioned before, the goal of that CRISPR reagent is to create a double standard break. And once they've done the double standard break, they've done their job. And now the rest of the work is done by the repair mechanisms of the plant. So when the host encounters a double standard break, it recruits a lot of enzymes to repair this double standard break. And this repair mechanism is often error-prone, and it creates deletions and insertions of DNA. And it's these deletions and insertions that disrupt the open reading frame of a gene. So if your target's in the open reading frame of a gene, you'll disrupt that gene, and you'll obtain a result. You'll have a loss-of-function gene, hopefully. Alternatively, you can cause a double-stranded break and then add a donor template. And this is called a more advanced genome engineering technique called genome editing or gene insertion or knock-in. And Tomasz is going to talk a little bit about this later on. So here are some really nice examples of genome engineering in plant pathology. It seems that plant pathology is a real beneficiary of this latest genome engineering or genome engineering reagents. So just this year, there's been several papers come out. So here's an example here in wheat, where they mutated the 3 homologs of the MLO gene in wheat, and they were able to get, so this is a single mutant here, these are the double mutants, this is the triple mutant here. You can see it has a good resistance to powdery mildew. A second example is this transcription factor ERF922, and they were able to delete that or create knockouts of that, and they were able to show this is the wild type, and these are examples of the transgenic knockouts, and they were able to get rice blast resistant plants. Another example is where they knocked out the elongation factor 4A gene, which is required for polyvirus transcription, and they were able to generate polyvirus resistant cucumber. So no virions at all were detected. Okay, so in my own research, I'm interested in small RNAs in legumes, and so I use the CRISPRs to do functional genomics. So to date, I've knocked out many components of the microRNA pathway in soybeans, such as DICER1, HEN1, RDR6, for example, DICER4. And so now I'm extending that using the CRISPRs. And so a target that we're interested in at the moment is this DRB2, which is a cofactor of DICER1 in the microRNA pathway. And Rees et al. Has recently shown when you knock this gene out in Arabidopsis, you get quite striking drought and salt tolerance. And so there's some mechanism here that DRB2 is involved in. So we're interested in seeing what that would look like in soybean, or looking at knocking out DRB2 in soybean. But because soybean is paleopolyploid, most of the genes are duplicated. So we had to knock out 2 of these genes. So we created a CRISPR that targets the first exon of both these DRB2 genes. So we put that into tissue culture, and we recovered 3 transformates from tissue culture, and 3 out of the 5 transformed plants had T0 mutations. We next confirmed heritable transmission of these mutations, and we were able to confirm that 2 of the 5 plants were T0 quadrillelic double mutants, and 1 was a T0 heterozygous double mutant, so wild type alleles for 2A and 2B, and also the mutants. And so you can see here the double mutants had a 7 base pair deletion, a 1 base pair deletion, a 4 base pair deletion, and a 7 base pair deletion. And these deletions knock out the reading frame of these plants. And so we have a double mutant now, a whole series of mutants that we can then do phenotypic analysis. The second application that I've been using the CRISPRs for is validation of GWAS candidates. And so we use genome-wide association studies to identify symbiotic nitrogen fixation candidates. Now, typically, GWAS is a great way of just generating a whole large candidate list, but very few studies go forward and validate those candidates that they identify by GWAS, because typically, in the past, it's been very difficult to generate these mutants. And so in this study, we were able to identify 10 candidate genes, and then very rapidly just engineer CRISPRs for these, and then put them into, transform them, and then get 10 candidate mutant genes of these, or mutants for these candidate genes. So very quickly, very rapid. So here's an example of one here. It just gives you an idea of the efficiency of the mutation. So this is the target site in the first exon, and this is the PCR assay. And you can see here if there's 3 bands, like 1, 2, 3, that indicates it's a heterozygous. If there's 2 lower bands here, it means it's wild type. And if you have this upper band up here, this is a T0 homozygous mutant. So in the T0, we're getting homozygous mutants very, very rapidly and very efficiently. And then you can sequence these and confirm them in the T1. And so for this line here, it's a biallelic homozygous mutant. It has 11 base pair deletion in one allele, and it has a 1 base insertion in the second allele. And then we can use these mutant lines rapidly or straight away to do a phenotypic analysis. And so we were able to use 2 mutational events to test nodulation, so using a robust sort of phenotyping assay to test nodule production in these lines, and we can show that they were significantly reduced in those mutants. The third example I wanted to quickly just talk about is the use of CRISPRs to complement plant breeding, especially plant breeding for stem rust resistance. And this is a project that I'm just starting now with the USDA. And so we want to combine these 2 stem rust resistance loci, SR31, which is effective to all the stem rust except UG99, and this stem rust resistant loci, 1RS amigo, which is effective to all stem rusts, including UG99, with the exception of 2 very non-related rusts, which I won't talk about. So we want to combine these 2 resistance loci into one plant to generate a plant that is resistant to a large range of stem rusts. And so the way we're doing it, so this resistant loci is from, or these resistant loci are from 2 distinct rye integration lines found in wheat. So this, for example, SR31 is from Petkus rye, and it is the small chromosome 1 of this Petkus rye that's been integrated into this Pavon wheat line. Similarly, this SR1RS is from Insave rye, and it's a chromosome integration into Nakota. So we did we crossed these lines to get the F1 heterozygous, then we top crossed it to Chinese Spring, and then we looked for recombinants that had both resistance loci. And so we've identified plants that have both these resistant loci. But the problem with these rye integrations is they have these seclin loci, this seclin locus, I should say. And so my role now is to remove this seclin locus. And this seclin locus is not wanted because it gives poor grain quality. The dough doesn't rise very well. And so we want to remove this. It's 190 kilobase per fragment in the wheat line. So we have 2 strategies to do that. The first strategy is to create a CRISPR reagent that has a target site on either end of the locus. And then we have PCR primers on 200 base pairs downstream and upstream of those target sites. And then we introduce that into the plant by plant transformation, and then we can screen T0 plants by just simply this PCR assay. And if we can get a 400 base pair, approximately 400 base pair fragment, that's indicative that that is being deleted. So that's what we're hoping to do with that. The second strategy is to use a 4-plex CRISPR that targets multiple regions of this locus. This is a very highly competitive, very similar gene. There's 17 seclins, and we have target sites all throughout that region, and then we're going to use the same primers to sort of see if we can identify mutants that way. But really, the goal is to just remove that locus from our wheat lines, and then we can start doing other gene editing approaches. So this latest multiplex CRISPR thing is a little different to the ones I just previously started off talking about. This is the one that Tom Ash is going to talk to you about now. So I'm just going to stop there and let him tell you the story about his fancy new multiplex CRISPR reagents. Oh, I should also mention that rather than ask questions, we'll have a Q&A at the end of the talk. I forgot to mention that. Sorry.
Okay, good morning. Thanks for the introduction, Shaun, and thanks for inviting me to speak here. And so Shaun showed you in his presentation the basics of the CRISPR-Cas9 biology, and he showed you some examples of gene knockouts, how to do gene knockouts with CRISPR-Cas9. And I want to go beyond just gene knockouts, and I want to talk about this platform or tool set that we developed in the Voitage lab for plant genome engineering that can be used for both the basic and more advanced genome engineering applications. So here's just a few examples of plants in which genome editing was used to either study gene function or for crop improvement. And I'm showing this because I want to stress out that it was mainly possible because, or thanks to these two types of programmable nucleases, talents, and mainly CRISPR-Cas9, that is really made it easy to introduce targeted double-strand breaks in basically any plant that can be genetically transformed. So this wouldn't be possible only like 3 or 4 years ago, and it's only possible because we have these nucleases now. And I want to show this slide that Shaun already showed, just because it's important to say that most of the genome editing approaches that we have rely on endogenous DNA repair pathway to make gene modifications. So when we introduce a double-strand break, it can be repaired, or most of the times it is repaired by this non-homologous end joining pathway, which is basically just rejoining of the broken ends together, and it's error-prone, so that's why you can get short insertions or mutations at the target site, and you get, oftentimes you get loss of gene function. But then on the other hand, we also have this homologous recombination pathway that uses a DNA donor template to copy information and kind of seal the break. So if you provide this DNA donor template that carries your desired modification, you can get the HR pathway to incorporate that modification into the side of the break. And this strategy is called gene targeting, and really is much more challenging than just introducing these simple knockouts, just because this HR pathway is not the preferred DNA repair pathway in plants. Now, this is the most common gene editing approach just because of what I just said. This is introducing gene knockouts using CRISPR-Cas9. CRISPR-Cas9 because it's the most easily engineerable nuclease platform, and targeted gene knockouts just because this is most of the times you get gene knockout from introducing a targeted double-strand break in a gene. But really, we can do much more using CRISPR-Cas9 and talents. And some of these other types of modifications might be necessary in case you want to do, for example, gene stacking or creating new alleles of genes, or simply you just want to target more than one gene. You want to do multiplex targeting modification in order to do more advanced metabolic engineering or even studying QTLs. And also there is a few methods that have been described that can be used to further increase or tweak the genome editing outcomes, but are not yet widely used. And this is because people might require more expertise and, more importantly, new tools that might not be easily available or straightforward to use to practice these other types of gene editing. So to make this, make combining and testing different genome engineering approaches possible, we decided to integrate all of these tools into one comprehensive system. And this is a vector tool set. It consists of 2 different vector sets, actually. One that we call a direct set, simply because all the specificity determining elements, by which I mean either the guide RNAs or the TAL repeats, are cloned directly into a transformation backbone, which is a tDNA or a non-tDNA vector that is transformed into plant tissue. And this is to be done in as few cloning steps as possible. So usually for the Cas9-based constructs, it's only a single cloning step. So this is the fastest approach. On the other hand, we have a modular set here that contains all the functional elements, such as the Cas9 expression casnets, TALIN casnets, GFP, visual markers, and other genes in separate modules And these modules are then combined using a simple Golden Gate cloning protocol into the final transformation backbone. That is, again, either tDNA or non-tDNA vector. So, and this is how their modular system is organized. We have 3 modules, A, B, and C. And the module A, these are usually pre-made. They contain the Cas9, different types of Cas9 expression cassettes, including Cas9 nucleases, nicases, DAT enzymes, even transcriptional activators. And then the module B contains a cloning cassette to insert one or multiple guide RNAs, or it can be a TALEN. And module C contains more guide RNA cloning cassettes. It can also contain different gene cassettes such as GFP, and it can also accommodate the donor templates for gene targeting. And so once the guide RNA repeat or TALEN repeats are cloned into the module B and donor templates into module C, the 3 modules are then assembled in a second cloning step into this transformation backbone. And in addition to tDNA and non-tDNA, we also have transformation backbones that contain viral replicons, which we'll talk about shortly. And on the other hand, if you just really want to do simple gene knockouts, this is the easier way. This is the structure of the direct vector, where you don't have to combine anything. You simply insert your guide RNA spacer sequence into this vector that already has all the other elements necessary for genome engineering. It has the Cas9 expression cassette, it has the selection marker, and so this is done in one step. It's ready to use. Now, there's a lot of applications of this system, and I don't have the time to talk about all of them, but I just want to go briefly through these 3. And one of the most exciting applications of the system is obviously multiplex gene editing with a CRISPR-Cas9 system. And until now, this has been usually done by combining the Cas9 enzyme, or the expression cassette for the Cas9 enzyme, with individual guide RNAs, multiple guide RNAs, and each of them was usually expressed from a separate gene cassette or guide RNA cassette expressed from a separate polymerase III promoter, like the U6 promoter in this case. And while this approach is functional, constructing and propagating such vectors might be problematic due to these long repeats of the promoter repeats and also the large size of the construct. But there have been at least three other methods described for production of individual guide RNAs from a single transcription unit or single transcript. One of them uses this ribonuclease that is called CSY4, and then that recognizes and cleaves 20 base pair RNA hairpins. The other one uses endogenous enzymes that usually process the tRNA sequences in the plant cell. And the last one uses a ribozyme, a self-cleaving ribozyme, to release these individual guide RNAs from the single transcript. So if you place these recognition sites for either of these 3 types of enzymes in between the guide RNAs in the single transcript, you can get the guide RNAs released from the transcript. And that whole transcript can be driven by any type of promoter, such as the polymerase II promoter, like in this case. So we included all of these systems, all of these expression systems in our cloning set, and we designed this experiment in which we had 2 guide RNAs that are shown here. They are called here guide RNA 9 and guide RNA 10. And we also added this more commonly used technique where you express each of them from a single polymerase III promoter, either 2 identical or 2 different promoters. And we tested all these constructs because we didn't know really which one of these is the most effective one in plants, really. So CSY4 has been first described in mammalian cells and has been shown to be very effective, but has not been tested in plants. On the other hand, the tRNA has been first described in plants and has been shown also very effective, but we didn't know how they compare in terms of efficiency and how they compare to this other approach. So we designed that experiment with the 2 guide RNAs that are targeting 2 sites in a single tomato gene, and these 2 sites are 85 base pairs away. So in the case both of these guide RNAs are processed correctly and are functional, we can expect all of these gene editing outcomes, and that's including a long deletion between the two guide RNA sites, short indels at either or both of the sites, inversions, or even insertions. And so we took these two guide RNAs, placed them in all of these expression constructs using our cloning system, and we made a single vector that we transformed into tomato protoplast here. And then 48 hours later, we extracted the DNA and we deep sequenced the target locus. And we could see, we're very happy to see that both of these CSY4 and tRNA multiplex processing systems were very efficient in making mutations. Actually, about more than 40% of all the reads had some mutations in them. But what was even more surprising was that these were almost twice as efficient as any of the three constructs that had the guide RNAs expressed from the individual polymerase III promoter. So this was a really good thing to see. And then next we decided to go, decided to see if we can express more than just two guide RNAs, and if this is functional also in other plant species. So here we expressed 2, 6, or 12 guide RNAs, a pair of guide RNAs always targeting a single gene for deletion in either barley, wheat, or tomato protoplasts. And really, we could see that in all of the genes that we targeted, in all of these species, we could always detect these long deletions between the two guide RNA sites by simple PCR and sequencing. So this shows that really the system is functional. And here we use the, I forgot to say, here we use the most efficient CSY4 system that we identified here was the most efficient. So this shows that really the CSY4 system is capable of correctly processing up to 12 guide RNAs from a single construct and introducing mutations at up to 12 sites in the genome, and in this case up to 6 genes. We can target up to 6 genes for deletion. But what this doesn't show is that this can be done in a single cell, since this is a population of cells. These are protoplasts. And so to show that, we need to go to a whole plant. And our collaborators at the University of Sao Paulo had been working with this wild tomato species that has a very good resistance to insects and to also to high salt concentrations. But the genes underlying this resistance haven't been described yet. So they had this idea, why instead of bringing this or harnessing this resistance by breeding it into a commercial tomato variety, why don't we just do the opposite? Why don't we molecularly domesticate this wild tomato species? And by that, they meant basically increasing the size of the fruits, the number of the fruits, improving the color, making the plant or inducing the terminate growth. And really, tomato is a very good model for studying domestication. And since it is believed that all of these genes have been well described, and it is believed that there's only about 10 genes, loss of function mutations of which have major impact on fruit morphology in the domesticated tomato. So we picked 6 of these genes that are shown here, and we targeted this for deletion using our CSY4 multiplex editing system. We are hoping to get something like this, or something that looks more like a cherry tomato than like this weedy looking plant. And so yeah, we had very high expectations, and there might be a long way to that, it might not be possible, but at least we could test our system in a plant, in a whole plant. So this time we had 6 guide RNAs, each one of them targeting one single gene, one of the 6 genes. And then on the same construct, we had the Cas9 and the CSY4 gene for processing that guide RNA transcript. And so we transformed this into tomato, the wild tomato, and we were happy to see that already in the T0 generation, we could see phenotypes for some of these genes. And these are loss of function mutations, so you have to have a homozygous mutants to show the phenotype. Like this one, this mutant here has determinate growth, for example. So that was very encouraging, and we were really excited to see the T1 generation out of these T0 plants, and we screened this molecularly by PCR and sequencing, and it was very nice that in the first 4 of these 6 genes, all of the plants that we screened from a single transgenic line, we found mutations, either in heterozygous, which is the yellow, or homozygous state, which is the red. So there's not a single one that didn't have a mutation. Then the last gene had only a base substitution, which we still have to figure out if that comes from gene editing or is just a SNP. We didn't find any mutations in the gene 5, but this is just the first experiment we did. And we are really excited that it works so well, at least for the 4 out of the 6 genes. And so this showed that, yeah, at least up to 4 guide RNAs can be functional in the same plant. In the same cell, you can make a plant with the mutations in 4 genes at the same time. And so we have used this. We've introduced multiple gene mutations in other species as well, including Medicago truncatula, including long deletions up to 60 kb, Setaria and cassava, and we're working on implementing this system also in other crop species. So another application of the system could be, or as an alternative to introducing multiple mutations with multiple guide RNAs, or targeting genes for deletion with 2 guide RNAs, we can enhance the rates of mutagenesis or with just using a single guide RNA and adding an exonuclease, an expression cassette for an exonuclease. And this exonuclease would resect the broken ends after the double-strand break is made and increase the size of the deletion, and that way also increase the chance you'll get a knockout mutation. And for this, we picked this exonuclease called TREX2, which has been shown to be effective in mammalian cells. And so we included it in our cloning system in this module that has the Cas9, and the TREX2 is fused to the Cas9 gene by this P2A sequence. And so we tested this with a single guide RNA on a few sites in tomato and barley protoplasts. And this is called a T7E1 assay, and I won't go into details. Basically what you see here, the digestion products here, the intensity of the bands from the digestion products, it basically tells you how many mutations you have. And we could see that when we added TREX2, we always get slightly more mutations than when we didn't add TREX2. It was about a 2.5-fold increase in the mutation rate, but also in mutation deletion size, deletion length. And for example, in this experiment with barley, we could see that 100% of the clones that we sequenced had mutations longer than 10 base pairs, compared to 0 of such mutations in the case where we didn't use TREX2. So really this shows that TREX2 is a good alternative, it's an efficient tool to enhance mutagenicity of the Cas9 editing. And at the end, I want to spend just a few minutes talking about precise genome editing and using Gemini viruses, Gemini virus replicons for doing precise genome edits in plant genomes. And by precise genome edits, I mean user-specified edits, since when we do gene knockouts, we can target them where we don't know exactly what type of mutations we get at the end. But here, when we want to do a user-specified, for example, an amino acid substitution in a gene, we most of the times have to rely, as I said in the beginning, on the homologous recombination pathway that uses that DNA donor template to copy that information and seal the break. So we provide the donor DNA template. With our desired modification, we introduce the double-strand break in the gene of interest, and based on the homology of the donor template to that side of the break, that gets incorporated. So, or we can hope that it will get incorporated, but that doesn't happen very often, just because the NAG, most of the breaks are repaired by the non-homologous end joining. And so that's why there have been only a few successful cases where gene targeting was used in plants. And if you want to increase the efficiency of it, we need to look for ways to stimulate the HR, the homologous recombination. And this is one of them. The whole idea behind using Gemini viruses for gene targeting in plants lies within amplification of the donor template within the plant cells into many hundreds or thousands of copies, which would basically make it better available for the HR machinery, and it would increase the chance of it being used as the template for the repair. So Gemini viruses are single-stranded circular DNA viruses. Here on the left, this is the structure of a native bean yellow dwarf virus, which infects a wide variety of plant species. And it has these code and movement proteins that are essential for movement of the virus from cell to cell. It has the replicase proteins that are necessary for replication, and also has these non-coding long and short intergenic regions that serve as promoters and terminators. And to make this a vehicle for amplification of the donor template and the nuclease for genome engineering, we removed the movement and code proteins. So this is not a virus anymore, but it's still a replicon. It still can replicate because it still has these replicase proteins. And so this is how it works. You place your nuclease and your donor template on a replicon like that. So it is flanked by these two long intergenic viral regions that are recognized by the viral replicase. You can place it on a tDNA, and then when it gets delivered by, for example, agrobacterium in the plant cell, here the replicon gets released from the tDNA, it gets circularized, and it replicates into many hundreds or thousands of copies, and that in turn can induce gene targeting. And so we have been able to show that this approach is functional in tobacco and tomato. So in this proof of concept experiment in tobacco, we delivered this donor template here. Well, actually, it's here. This is the replicon with the donor template that contains parts of the GUS and NPT2 genes that are designed to complement a broken GUS NPT2 transgene in tobacco. And so we delivered this on the replicon, all the regions on the replicon except for the viral replicase that was delivered on a separate tDNA construct, and we agro-infiltrated tobacco leaves. And the readout was the number of blue spots resulting from GUS activity, from reconstituted GUS, because the GUS is, in these transgenic tobacco lines is broken, it's not functional, and we can only see activity when it gets repaired by the replicon. And we could see that when we co-delivered the replicase with the replicon, we could see increase in gene targeting frequency from 10 up to almost 100-fold compared to when we didn't deliver the replicase, so the non-replicating control. So this was done transiently in leaves, but then we decided to target an endogenous gene in tomato. And here, this gene is called ANT1, or anthocyanin 1, which is a transcription factor whose overexpression leads to accumulation of anthocyanins and purple coloration. And so in this case, we had this donor template that contained a strong 35S promoter in addition to an NPT2 selection cassette, and we were inserting this sequence in front of the ANT gene in the genome with the help of a TALEN-induced break. And this was, in this case, everything was on a single construct, on a single replicon, including the viral replicase, and that we delivered into plants by agrobacterium, and we succeeded with our goal. You can see the resulting plants here with the targeted insertion. They are purple, dark purple compared to the wild type. And the frequency with which we could get this purple tissue was significantly higher, again, compared with the non-replicating regular tDNA control. We also were able to recover progeny from these plants to show that the targeted insertion is heritable. And we also could find or screen out the homozygous insertion mutants in the T1 by PCR. But perhaps the most interesting finding that we had from this came from a southern blot and PCR analysis of some of these major plants, and which showed that there was no additional random tDNA insertion, because the replicon is delivered on a tDNA, so the tDNA can insert randomly in the genome, in addition to the target insertion of this sequence. So there was no random insertion of tDNA in these plants, which suggests that the insertion of the tDNA is not necessary or not essential for inducing gene targeting using Gemini virus replicons. And so Gemini virus replicons are probably one of the best approaches for gene targeting in plants to date. And so I talked about the applications of the system and showed you some examples of what it can be used for. I also want to mention we also developed this set of online tools to streamline vector selection and primer design. Basically, you can select the type of vector you want, type of the genome engineering reagent, selection marker, promoter, and so on. And by this step-by-step selection process, and it will tell you which vector is the appropriate one that you want to use. And a similar tool for primer design. This tool designs primers that you need to construct the multi-guide RNA constructs that I just showed you. And I need to acknowledge all the people who helped this massive effort of putting all these regions together and testing them. And I'll thank you for attention, and I'll let Diana come tell you about her experience.
Thanks, Tomas. So our next speaker is Diana Trujillo, and Diana is a graduate student in Nevin Young's lab. She's a bioinformatics graduate student, and she decided to get off the armchair and hit the bench, and she's got a fantastic story to tell here, and I guess her story shows you how very approachable these tools are to everyone. You just pick them up and make mutants. It's fantastic. So thank you.
All right. Can everyone hear me? Yep. All right. Thank you, Shaun, for inviting me, and thank you, everyone, for attending. My research has focused on small signaling peptides in Medicago, which is our model legume species. And so I should probably give you a little bit of background on why I am interested in Medicago. And basically, it's the site of, or legumes are the site of symbiotic nitrogen fixation, which occurs in this relationship with rhizobial bacteria in the soil. Basically, this process involves signaling peptides, which facilitates communication between the rhizobial bacteria in the soil and the legume host. And so these signaling peptides allow the rapid evolution of different nodule morphologies and rhizobial morphologies, which can determine the efficiency of nitrogen fixation. And so the specific aspect about these signaling peptides that I'm interested in is lineage-specific expansions of gene families within or across different species in the legume family. And so one of the examples of this is the NCRs, which in soybean, let me see. Yeah, which in soybean, there's at most one member of this NCR gene family, whereas in Medicago, which is the model legume that I'm interested in, there's over 600 identified members of this gene family. And so this leads to changes such as the morphology of the rhizobia is actually determined by these cysteine-rich peptides in Medicago. And so I wanted to investigate whether I could find more of these lineage-specific expansions in legume species, and specifically in Medicago. And so I developed this pipeline to basically, in summary, identify gene families that are nodulation-related and that have a signaling function. And I did this just by biasing my data with nodule RNA-seq data and by filtering transcripts to only those that have signal peptides. And so after tweaking my pipeline, I had a bunch of candidate gene families, and kind of just to lead my project to from the bioinformatic process to actually showing that there is a biological function behind these families that I've identified, I chose one of them for further study, and this is a gene family called the embryo-specific proteins, but as you can see here, they're probably badly named because there are actually subsets of this gene family which are overexpressed in nodules. And so this gene family expanded from 1 member in soybean to 5 in Medicago, and my ultimate goal was to try to knock out as many of these as possible, because going in, I didn't know if these proteins were redundant or not. And so in order to do that, 1 year ago, I decided to jump back into the lab and use this CRISPR technology. And so I adopted different strategies to try to target all 5 of these nodule-related ESPs, or at least as many as possible. And so the first strategy is what Tomasz has talked about, which is basically the multiplex expression of guide RNAs. So trying to knock out all 5 of them requires having having multiple guide RNAs to target multiple genes. And so another strategy that I used was to try to make these guide RNAs flexible in that, for example, I have guide RNA 1 is able to target ESP gene 2, 3, 4, and 5. And this is simply due to the homology between the high sequence homology between the ESP genes. And so actually, there's a mismatch in this target site, and so the other objective was to see whether the mismatches in the 20 base pair target site would hinder the efficiency of these guide RNAs. And then finally, the last strategy, which to me was the most desired outcome, would be to just knock them all out in one go through a large-scale chromosomal deletion. And so kind of to take you through the nitty-gritty of what this process implies at the end of your transformation process, you'll have, at least for my case, I had to screen each gene independently for every single one of my mutant lines. And so basically you have, after doing PCR of the region, you have a wild-type band. 2 bands would mean that the plant was heterozygous at that spot for deletion, or you could have a band of smaller size, which means that your plant is homozygous for the deletion. And like Shaun mentioned before, the other thing is you can just do a PCR from one end of your region to the other end of the region, and any band that shows up points to the fact that something has gone on, whether it's a 20 kb deletion or just spurious PCR. You have to confirm that through Sanger sequencing. And so this chart kind of just shows the results from the first strategy, which is the multiplex expression of the guide RNAs. And so these are 3 different vectors that I used. And so Tomasz talked about them, but the first one is using multiple promoters who drive the expression of each individual guide RNA. The second box corresponds to the CSY4 system, and the third box corresponds to the tRNA system. And so the sample size is kind of small, but you can see that the CSY4 system worked really well for me. And kind of what I would say is the takeaway point from this is that if you're going to go through the transformation pipeline anyway, it might be good to just use several different vectors and see which one works best depending on what your goals are. Especially if your goal is to knock out more than one gene, you might want to choose one that has the highest efficiency so that there's less screening to do later on. And so this is a representation of the mutants that I have obtained. And so these are ESP genes 2, 3, 4, 5, and 6 in that tandem duplication region. And so this is kind of just showing you that I was able to obtain the 5-gene knockout both through the large-scale chromosomal deletion as well as more mid-scale deletions combined with indels at the remaining genes. And something else that I'm really excited about is the fact that I also have 4, 3, 2, and 1 gene knockout lines, which will in the future allow me to do gene dosage studies, just to compare between the different lines and see if the number of these genes is important in nodule morphology. And so this kind of just shows that the strategy of going for large-scale chromosomal deletions works, and it's really useful if the genes you're interested in are within the same region. And my final strategy, which was to use flexible guide RNAs, was not as successful. And so if you're trying to target as many genes as possible, I would say maybe just try to use multiple guide RNAs that have 100% homology. And so this is results from my Sanger sequencing of each of the individual genes. And so when there was 100% homology, I had a higher number of lines with the indels at the expected location. Whereas, for example, here there's a mismatch in within this 20 base pair target region, and only one of 8 lines that I screened had an indel at that region. And so just to take it back to the biology, when we compare the wild type plants to the 5-gene knockout plant, this is kind of just a pilot experiment, I basically found that nitrogen fixation is not occurring as expected. The nodules are white and small compared to the wild-type nodules, which are pink and elongated. And basically what a white nodule means is that the plant is not expressing like hemoglobin, which is required for nitrogen fixation. And this is another experiment where I used different rhizobial reporter mutants that kind of just signal at what stage the rhizobia are still alive. And so this, if you get the blue stain, it means that the rhizobia are still alive at the infection stage. In the second column, they're still alive in the entry to symbiosome stage, so when they're actually invading the cells of legumes. And then finally, there's a lack of blue here in this final stage, which is the nitrogen fixation stage. And so that basically confirms the fact that this 5-gene knockout has deficiencies in nitrogen fixation. And so some summaries, some insights from the work that I've done is that if you have small gene families that you're interested in, and before you haven't been able to knock them all out at once, or it would have taken you 10 years to do, well, now you could do this within a year, because I guess depending on the number of genes you have, but I started about June of last year, and I already have phenotyping results, so that's very encouraging. Another insight is that I was able to obtain the 5-gene knockout plants, but I also obtained a range of other mutant lines, which will allow me to do further gene dosage studies. And just back to my original system, the disruption of all 5 of these nodule-specific ESP genes caused deficiencies in nodule formation and loss of nitrogen fixation ability. And with that, I would like to thank Shaun Curtin for inviting me here, but also because he was my mentor throughout my whole CRISPR adventures in the Nevin Young lab. And also, I would like to thank Tomasz for basically providing me with these vectors and helping me through this. And with that, I think we could probably all come over here and start taking questions.
Diana. That's great. So we have about 10 to 15 minutes now for some questions. If anyone has any questions to ask, now would be the time to do so.
Okay, let me get the name of the first speaker again.
Shaun.
Troy?
Shaun.
Shaun. Okay, on your GFP recorders, I was curious if you've done other colors. The GFP is like not real good at plants, but like have you done any with like any other colors?
So I think you have DS Red and do you have CFP? In protoplasts, the GFP works very nicely for. So what GFP are you using?
One jellyfish. We've used 3 different regions of it, but I don't work on bugs, and the problem is you get a lot of background.
Yeah, I mean that's.
And they're coming out with all these new colors. I didn't know if you guys were just doing that.
So we haven't. I mean, they could be very easily integrated into the system. So like
Right.
P Neptune and Honey and DS Red and PT Tomato or stuff like that. So we do have them, and yeah, that could be very easily integrated into your multiplex system quite nicely. Yeah, but it hasn't been done yet.
We see that you can knock out the gene or increase the genome of the gene and do that with CRISPR.
Increasing what?
Increasing the copy number of the gene.
Copy number.
Okay, so we thought about, so in soybean we were trying to do that with the cis nematode resistance genes. What we thought, we would try and delete them, and then we were hoping to see some sort of rearrangements where you deleted a large region and then somehow, I guess it was more of a pipe dream than anything. We never found them, but there has been efforts to do that, but they're largely been unsuccessful. But I think if you could use exonucleases, so to understand, like if you had a large deletion, say an ESP deletion, and that's floating around, there's potential for that to insert somewhere or catenate. So you would have 10 ESP genes. I mean, we haven't seen that, but we really haven't looked for it. And so that might require purposely looking for it. And so there may have been plants that we've overlooked that had that, like say 10 ESP genes. But to date, we haven't looked for it.
Hello, my question is for Diana. So you had the nodulation phenotype for your mutants for all the 5 genes. And although you have the nodulation phenotype, like you have a defective nodule, you still see the NIFH and BACA expression in them, like it's bluish. Did you do acetylene reduction assay on those nodules?
We haven't. We have been talking about it, kind of just to drive that point home that nitrogen
Okay.
deficiency is.
Because the presence of NIFH expression suggests that probably the nitrogen fixation is still going on in those deformed nodules, correct?
Well, the lack of
Yeah.
expression in the. In the
Deanna, can you come?
Sorry. So the lack of expression of the NIFH genes would suggest that those nitrogen fixation genes are not being turned on.
Okay.
Was that
Yeah,
the question?
but I see the expression in both normal functional nodules as well as in the defective one or the abnormal nodule. So that's why I'm asking, like, did you try the acetylene reduction?
We
assay
have
on
not,
them?
but that is something that we're thinking about doing.
Awesome. Thank
Yes.
We would like to apply the technique to fungi, in plant pathology to knock out some virulence genes. And the colleagues that I've talked to have tried and have been unsuccessful. And then it seems that with plants, the spectrum of plants that you can transform is limitless. So is there any reason that fungi would be less amenable to this transformation? And I was wondering, you showed a very nice pipelines where you can go online and pick different things. Would it be developed for transforming fungi as well?
Well, I think that might be a problem of the specific promoters or expression. since CRISPR-Cas9 has been shown to be functional in fungi. There's actually a fungus, a mushroom that was made with CRISPR-Cas9, it's genome edited. So I think it might be, you might need some specialized promoter maybe for expression in fungi, which can be incorporated into the system. We don't have them now. We have the plant promoters, but a Cas9 as a nuclease should be functional in fungi. And I think the CSY or the multiplex systems as well, since we tested them, they've been tested in many plants and animal systems as well. So I don't see a reason why they wouldn't be functional in fungi.
I had a question about the Gemini virus-based system.
Mm-hmm.
You said that you delivered it through agrobacterium tDNA, but you did not see any insertion of the tDNA in the genome?
That's correct.
Because I always thought that if you have a left border and right border on the tDNA, would integrate into the chromosomal genome?
Doesn't always need to integrate. You can get transient expression, right? And the only thing you need is to deliver the replicon to the cell. And once it gets released from the tDNA is no longer necessary. So whether it inserts or not, it doesn't matter for the replicon. The replicon still can replicate and induce.
But it could still insert in a
Yes.
exon somewhere in the genome, right?
It could, yes. It's possible. It just didn't happen.
Sorry, just to answer that question about the fungus, I'm pretty certain that the 35S Cas9 Cis4, which is driven by the Cestrum virus promoter, will work in a number of different fungal species. I guess it depends on what sort of fungal species. Are you working on biotrophs? That may be a little difficulty getting the transfection of that, but if you're using a plant U6 promoter, that's probably not going to work so well. But certainly the Cestrum virus promoter, part of your kit with the 35S Cas9, I think will work very nice.
And these polymerase III promoters that people usually use to express the guide RNAs, they're kind of specific to even the plant species. If a certain promoter is isolated, let's say from Arabidopsis, it will be more efficient in Arabidopsis than in soybean, for example. So that might be a problem if you have a polymerase III promoter from a plant and use it in a fungus, it might not work. Right? But these, in the multiplex system, we use polymerase II promoters that are kind of more or less specific for certain species.
On your stable transformation on the T1, how efficient you can get rid of the backbone sequences that you don't want to, like Cas9 or other tDNA sequences?
So what I've seen in soybean and metacargo, it's pretty routine. You look at the T1 and you screen you screen a collection of plants, and
what percentage of the plant?
So for that example I showed you, for number 3, I think I screened maybe 8 plants, and 2 of them were non-transgenic. But then we have sorry, I don't have a specific percentage, because I just I identified the non-transgenic plant, and then that's all I'm really interested in. Sometimes you do see events where you don't have, you don't find a non-transgenic or removal of the transgene. When you test, say, 20 T1 plants, you don't see anything yet, and that may be because there's multiple insertions. So occasionally you do get multiple insertions, but oftentimes you do get single insertions that can be removed. And so I've seen that in removal of zinc fingers from soybean, removal of talons from soybean, and now removal of CRISPRs from soybean and metacargo. So it's typically generally the norm. I don't know, have you observed removal of the transgene in your CRISPR? Oh, you're going to check that. So yeah, but I think it's pretty routine.
Hi, I have a question about deletions that were unintended off-target effects. Have you observed many? Do you have an idea of the rate of off-target deletions? You showed us some very successful targeted deletions. And how about off-target deletions?
Right, we haven't really checked for off-target deletions with the multiplex system, since we have never had any problems with toxicity. Like you can have toxicity with zinc fingers that can be inhibited growth or other phenotypes. We didn't see any phenotypes that would mean that there is something wrong. And we didn't check for that. We did check for off-target mutations with talents, but that's something else, right? So, and we didn't see any. It's usually not a problem with the off-targets, but yeah, we did not check.
And I should say, I have seen off targets, but in my vectors, but that's because I guess a little recklessly designed the guide RNA actually could hit another gene, and so I was able to get unintentionally. So it comes down to when you're designing your guide RNAs, make sure that if you have 100% off target, you're probably going to delete that as well. So it's important if you just want to delete one gene, make sure it's not going to hit anywhere else. So.
There's only a very few studies that actually looked at off-targets in plants specifically, and they have contradictory results. Some of them found some off-targets, some of them didn't, so we still don't know what the rules are exactly. Basically, you want that seed region of the guide in the first 12 nucleotides or so to not match any other sites in the genome, and in that case should be probably fine.
Hi, you mentioned that one of the species you used to knock out was cassava. Do you know which genes in cassava you were knocking out?
So in cassava, we are actually doing, we are not doing gene knockouts, we are doing gene targeting, we are doing gene replacement. And yeah, we have been successful. And we also, yeah, when we first tested the nuclease that we used for the gene targeting, 2 guide RNAs to delete a fragment from a gene and replace it with another one, we could get both the replacement and the deletion between the 2 sites. So it is functional in cassava as well.
So what was the phenotype you were looking at?
Phenotype of, so we are trying to create a herbicide-tolerant cassava. So,
thank you.
The use of the CRISPR technology in industry is very limited because there are a lot of legal issues that are mandatory that has to be fulfilled. And I'm just curious about if the USDA or the universities have to go through all these legal issues in order to use this technology. And I noticed that you are even you can use it for creating resistant varieties, but once you have the resistant variety, can you deliver that variety without facing those legal issues for using the technology?
Yeah, I think it's complicated with the legal issues since we still don't know where, even if you wanted to license it, where to go to license it. But as long as you are a non-profit, a university, or any non-profit organization, you can certainly use it. No one will tell you anything. Once you have a product and you want to sell it, then that might be a problem. Then there might be a legal issue, but no one knows yet.
Okay, thank you. And what about the USDA? I'm curious about the USDA as well, how it works.
Maybe Shaun can tell me.
So, yeah, I mean, it's sort of up in the air at the moment. No one really knows what's going to happen. So, for example, I'm doing gene targeting in wheat at the moment, in wheat notes, and I'm using that, I don't know if people are familiar with the Japan tobacco transformation protocol that was published in Agrobacterium Protocols. And that's got a pretty hefty license fee for that. So if it's for academic purposes, I'm fine to use that, to use it. But if I want to commercialize it, I might have a hefty fee ahead of me. I don't know how it's going to work. So now, but for the CRISPRs as well, we don't. No one knows yet. It's in the courts, and it might be for another few years.
Yeah. Okay. Thank you.
Personally, I think they should just let us do whatever we want, but
yeah.
It's not going to happen.
Can I add a comment to that question? I work for the USDA Biotechnology Regulatory Services, and if you have a question, if you don't know if your product is regulated, there is a process by which you can submit an "Am I regulated?" letter. And you can submit that letter and it will be analyzed and you will get a response whether your product is regulated or not. Because we are really confined by what the regulations and the law says. So nobody wants to stop research, but if you have a question, if you don't know, just submit a letter and we can answer that.
Thank you.
Thank you.
Yeah.
So my question is for Thomas. Regarding the plasmids, are they available? Where? And also the online page, can you give an address?
So we are trying to, we will make this toolset available on AdGene soon. It's not there yet, but we really hope it will be there soon. So right now we can distribute a limited number of these kits directly from our lab. And the website, you could email me and I'll send you a link to the website.
Thanks.
It's a general question. I would like to know your opinion about the use of CRISPR for mitochondrial and plastid DNA editing, the feasibility and everything.
So that's more a question of the delivery to mitochondria and protoplast, but certainly it's effective or it's efficient anywhere, I would say.
Do we know if there are already DNA repair system like nucleus and mitochondrial and plastid in order to enable this CRISPR-mediated gene editing?
Sorry, I didn't understand.
Are there already a DNA repair system available or present in mitochondrial and plastid organelles?
Well, we know that zinc fingers have been used, zinc finger nucleases have been used in mitochondria, so I don't see it, and that's based on always on the same DNA repair pathway, so I don't see a reason why CRISPR Cas9 wouldn't be active in mitochondria.
Okay.
But yeah, I have not done anything like that, so I can't really say.
Good, yeah, thanks.
So if there's no more questions, we can, one more. Sorry, can you repeat the question?
Limitation.
What is the limitation?
Well,
there are none.
Biggest limitation is the
transformation.
Transformation, the delivery of the reagents, right? You cannot really transform any plant yet, so.
So yeah, I mean, I think hopefully we're going to see some breakthrough improvements in transformation. That'll be a.
Use the general transformation process. Use the general transformation process.
Yeah, for the plants where you have transformation methods, it's really easy. There's not many limitations, but some other plants you can't really transform, so still have to look for better methods for delivery.
Okay, well, thank you very much for attending this session and seeing us to the end here, and thank you very much for all the questions. And,